whenever we run low in oxygen, what happens is that our metabolism is going to shift, right? It's going to shift from aerobic metabolism to anaerobic metabolism, right? That anaerobic metabolism is called fermentation. And actually, the reason why it's called fermentation, it's because pyruvate becomes the final electron acceptor as opposition to oxygen being the final electron acceptor. Okay? So, specifically, what happens? Well, during fermentation, we have anaerobic breakdown of glucose, right? So here, pyruvate is going to accept some electrons and be reduced into a molecule called lactic acid or lactate. This happens typically in animal cells, right? Like for instance, in your muscles, when you start exercising, you have this feeling of burning in your muscles. This burning is actually a high concentration of lactic acid, right? That accumulate and that cause you to have that feeling. Now, in animal cells, fermentation is going to happen in basically a burst of energy for muscles, but eventually the muscle will start running out of energy and start experiencing fatigue, right? Now, other organisms, including bacteria, have multiple ways of functioning without oxygen, right? But these multiple ways include lactate production, right? Fermentation, or other organic acids, not only lactic acid, but they can produce other organic acid. Some bacteria and some eukaryotic cells are going to produce alcohol, ethanol, for instance, right? And carbon dioxide. Now, when we consider the baking industry to make bread, to make croissants, all these good things that are good to eat, right? Yeast is very much what we use. When we use yeast for baking, the cake is able to take up volume because the yeast is consuming the sugar that we put in the dough, right? And that sugar is converted into alcohol and carbon dioxide. Carbon dioxide being a gas, when carbon dioxide is trying to escape from the dough, right, it causes the dough to rise. That's why you see the bread dough rising, right? Wine production, it's by the same process. Beer production, by the same process. And all of that, it's fermentation, okay? Where pyruvate is the final electron acceptor. So here you see an athlete who is feeling kind of tired here because of running low in ATP. Why does this guy run low in ATP? Well, he runs low in ATP because during fermentation, glucose is converted into lactate, right? Or alcohol with the release of carbon dioxide. Now, remember the two NADH that are produced during glycolysis. These two NADH are actually going to give off the electron to the pyruvate so that pyruvate can become lactate, right? Or alcohol and carbon dioxide, okay? So that at the end of the day, you have production of the coenzyme again, 2AD+, all right? Now, you have two ATPs that are invested. After hydrolysis, you have two ADPs, right? And these two ATPs are invested during glycolysis. Remember that, right? 4 ADPs are invested with 4 phosphate groups, right? That are attached to the substrate, right? The glyceraldehyde 3-phosphate that becomes 1,3-bisphosphoglycerate. Remember those, right? At the end of the day, you have production of 4 ATPs. But when we take a look at the overall outcome in terms of energy ATP of fermentation, we only have 2 net ATPs that are gained. So at the end of the day, when we exercise and that our body is not receiving as much oxygen as it needs, we undergo fermentation metabolism, right? Anaerobic metabolism. At that point, we only rely on glycolysis, so producing only 2 ATPs. Considering that though, how long can we keep exercising, relying on glycolysis to only produce 2 ATP each time for 1 molecule of glucose that is partially metabolized? Well, this is not sustainable. That's why when you exercise at some point, your body gives up because it cannot keep up with the demand in energy, right? So fermentation does not necessarily lead systematically to lactic acid production, right? It can also lead to alcohol production. That's basically how we produce alcohol in wine and beer, right? Now, from the very beginning, here we have a glucose molecule. That glucose is activated with 2 ATPs, right? Okay. Actually, I should point this one out, 2 ATPs, right? So activation, then we have pyruvate that is produced. We have our 2 net ATPs that are produced here, right? Okay. So now, after that, because of lack of oxygen, what is going to happen is that the NADHs here are going to give off their electrons to pyruvate so that there are 2 outcomes possible. One, it's lactic acid fermentation. The second option is the alcohol fermentation, right? So these are the 2 possibilities. Now, let me mention that some organisms, including bacteria, do not necessarily produce exclusively lactic acid. They can also produce other organic acids. Okay, just remember that. Now, we know that most of the time we are not tired. We walk, we run, we talk, right? We go about thinking and doing everyday tasks, and we don't feel exhausted, right? Because cellular respiration is going on all the time. And very importantly, because we are bringing in enough oxygen to sustain our activity, right? So now let's go inside the mitochondria to see actually how things come at play. First and foremost, let's take a look at the structure of the mitochondria. A mitochondrion has 2 membranes, an outer membrane and an inner membrane right here in yellow, right? Inside, you have a matrix. That's the opening right here. This is where many reactions are happening. Now, between the outer membrane and the inner membrane, the outer membrane right here and the inner membrane, you're going to have a space. That space is the intermembrane space. That intermembrane space is going to be essential for accumulating some chemicals, right? But let's close that parenthesis. The second aspect I would like to mention, it's the matrix. In the matrix, which is the open space inside of the mitochondrion, bunch of reactions are going to happen in there, right? Now, finally, but not least, I would like to point out to those structure the cristae, right? This is the folded inner membrane. In that inner membrane and the cristae, this is where we are going to have the electron transport chain occurring. Now, let me repeat that. The electron transport chain occurs in the inner membrane of the mitochondria, right? Okay, so please remember that. Now, let's take a look at the reactions themselves. First and foremost, the preparatory reaction or the transition step. That transition step is going to happen in presence of oxygen gas. All right. So in that transition step, specifically, what is going to happen is that pyruvate, remember that for one molecule of glucose, you have two pyruvates. I want you to remember that. So pyruvate is going to be oxidized. How is that? You have that NAD plus here is going to accept an electron from this pyruvate that is going to lose one bond here. Okay, when that bond is broken, we have release of a carbon dioxide, right? And as carbon dioxide is released, there is a 2-carbon molecule here that remains behind with the addition of CoA. Now this 2-carbon molecule is going to actually become what we call acetyl-CoA. acetyl-CoA, acetyl coenzyme A. But don't worry about that. So, and this happens within the matrix of the mitochondrion. That's a transition step. Now, let's move to the citric acid cycle. During the citric acid cycle, this is where a lot of the electron carriers are going to accept a bunch of electrons because the remainder of the carbon atoms are going to separate from each other at this point. Remember now, we went from a 6-carbon molecule to 3-carbon molecule. That was the pyruvate. Now the pyruvate has split and now we have acetyl-CoA, right? That acetyl-CoA, it's a 2-carbon molecule. Okay, so 2-carbon. Now the 2-carbon molecule is going to undergo a series of chemical reactions. In that chemical reaction, the first reaction consists of adding the 2-carbon molecule to a molecule here that is called oxaloacetate. Now, between you and I will not bug you with that name, okay? I will not bug you with that name, and I will not bug you with some intermediate of glycolysis, okay? I will come back to that so that for the sake of exam preparation, right, you don't overwhelm yourself with some aspect I will not bug you with, all right? I will come back to that. Okay, so you do have this 4-carbon molecule that is called oxaloacetate, and again, I will not bug you with that. That oxaloacetate combined with acetyl-CoA is going to form a 6-carbon molecule that is called citric acid. Okay, that citric acid is the reason why we call this sets of reactions citric acid cycle. Okay, so the first chemical is citric acid. That citric acid is going to undergo oxidation, and you have here production of a NADH, right? Another NADH that is produced here. I spare you the name of these chemicals, right? Because this is life on planet Earth, right? It's not a biochemistry class, so I'm not going to bug you with those details, right? Now, you have carbon dioxide that is lost here and carbon dioxide that is released here. At that point, because you have carbon dioxide released here and here, your acetyl-CoA molecule doesn't exist anymore, okay? So after the 2 carbons have been released into carbon dioxide, the 2 of them here, your glucose is gone, okay? So then we're going to have ATP that is produced here. Now, once ATP is produced, what is going to happen next with this 4-carbon molecule? It's that you're going to have regeneration of the oxaloacetate molecule, okay? Now, you start with oxaloacetate, you have to end with oxaloacetate. If you don't end with oxaloacetate, you don't have a cycle. All right, so you have to end with oxaloacetate. In the process of regenerating oxaloacetate, you have production of FADH2 here and NADH here. So that basically all the reactions that are going to happen. Now, what is the outcome of the citric acid cycle? In other words, what are the input and what are the output? Now, as input, after glycolysis, we start with 2-pyruvate, right? This 2-pyruvate, after the transition step, become 2-acetyl-CoA. So we have 2-acetyl-CoA. This 2-acetyl-CoA are going to release 4 carbon dioxide. Likewise, we're going to have 6 NAD plus that are going to produce 6 NADH, okay? And 2 FADs are going to produce 2 FADH2. In the process, 2 NAD are going to, 2 ADPs are going to accept 2 phosphate groups to form 2 ATPs. And the process by which ATP is produced here, it's similar to the process that was used during glycolysis, which is substrate level phosphorylation. Okay? And why substrate level phosphorylation? Because you have a molecule here, okay, that is going to transfer a phosphate to ADP so that phosphate can be produced. ATP can be produced, all right? Okay, so I want you to remember the input and the output of the citric acid cycle, okay? And for glycolysis, I also want you to remember the input and output of glycolysis. The intermediate, I will not bug you with those. I want you to remember the input and the output of glycolysis, all right? And also the input and the output of the transition step, all right? Let me close the parenthesis. Now let's move on. So now we are at the last step. The last step is the electron transport chain. That electron transport chain is going to happen in the inner membrane, and specifically in the cristae, because this is where the electron transport chain is located. That electron transport chain is going to be responsible for accepting all the electrons that were cut during glycolysis, 1, 2, the transition step, 3, the citric acid cycle. Okay, so all these electron carriers loaded with electrons that are highly unstable, very excited, containing bunch of energy, now are going to be given to specialized players in cellular respiration to handle that energy and convert that energy into ATP, right? So now we have 2 NADH produced here in glycolysis, 2 NADH here produced in the transition step, 6 NADH produced in the citric acid cycle, and 2 FADH2 produced there. And all of this through the electron transport chain are all going to contribute into ATP production. Now, here is what I'm not going to bug you with. I'm not going to ask you how many ATP, for instance, the 2 NADH from glycolysis contribute to for the overall production of ATP for one molecule of glucose that is broken down. I will not bug you with that, okay? That's too much. That would be too much detail. But what I want you to remember is that the electron transport chain produce as much as 34 or 32 ATPs. Let me repeat that. I want you to remember that the electron transport chain in the overall ATP production of cellular respiration contributes to 32 to 34 ATP molecules. So that at the end of the day, well, they are the major contributor, the electron transport chain, I mean. And notice that at the electron transport chain, this is where oxygen is going to come at play, accept these electrons, and oxygen becomes H2O, water, right? Now let's take a look at some details here. So we have here in the matrix, we have the NADHs, we have the FADH2, right? So these electron carriers are going to bring electrons, transfer these electrons to electron transporters here. These electron transporters are going to suck energy from the electron. As they suck energy from the electron, that energy is going to go toward activating the transporter so that the transporter can carry hydrogen ions from the matrix to the intermembrane space. I want you to remember that. Now these electrons have lost some energy, but they are not stable yet. They're going to be passed to another electron acceptor that is now going to pass the electron to another transporter. That transporter also is going to suck energy out of the electron. That energy that is sucked is going to fuel, activate the transporter so that the hydrogen ions are transported from the matrix to the intermembrane space. So now what you see, it's that the energy that was in the electrons is being used to power electron transport systems to transport hydrogens across the membrane, so in the intermembrane space. Now we are building up a gradient of concentration of hydrogen ion, right? So that we have some more carriers here, electron carriers that are going to transfer electron to another electron transport player here that again is going to suck energy from the electron, use that energy to transport hydrogen ions across the plasma membrane. I mean, across the membrane of the inner membrane of the mitochondrion. Now we have definitely an unequal distribution of hydrogen ions across the membrane. So now we have a pressure that is going to exist on this side toward this side, okay? In the intermembrane, pushing on the surface of the inner membrane, right? So now the question is, because these chemicals are charged, they cannot cross the plasma, they cannot cross this membrane, the inner membrane, I'm sorry. They cannot cross the inner membrane. In the meantime, right, the electrons have lost enough energy to be actually accepted by oxygen, right? And as oxygen accept electron, in reality, they accept hydrogen ions so that they can become water. Now, question is, how is this hydrogen ending up across, knowing that they cannot cross freely the plasma, I mean, not the plasma membrane, but rather the inner membrane of the mitochondria, where what happens is that there is another complex protein here embedded in the membrane. That complex protein is actually a channel. That channel let hydrogen ions, let hydrogen ions go through the system and come here in the matrix. These hydrogens now are the ones that are going to combine with hydrogen, with oxygen to form water. But let me close that parenthesis. Let me mention that the flow of hydrogen ion through is such that that flow represents energy, right? Remember, we are going from high concentration here, right, toward low concentration. And that flow represents kinetic energy. That kinetic energy is enough to power a domain here that is an enzyme. That enzyme is able, when active, to attach a phosphate to ADP so that ADP plus the phosphate here becomes ATP, right? Now, earlier, the phosphorylation that we saw in the citric acid cycle and glycolysis was substrate level phosphorylation because phosphate was taken from the substrate to add to ADP. Here, the origin of phosphate is different, right? And the reason why phosphorylation is happening is different. Here, you have what we call oxidative phosphorylation. That oxidative phosphorylation means that you actually have phosphorylation that is powered by the acceptation, or actually no, by the loss of electron. So NADH is releasing electrons. FADH2 is releasing electrons for the phosphorylation to happen here, okay? That loss of electron associated with ATP production is called oxidative phosphorylation, okay? Now, know that when it comes to the overall metabolism and the yield of ATP for one molecule from one molecule of glucose, in glycolysis, remember that we have two NADH that are produced, right? Two ATPs, net ATPs that are produced. In the preparation step, you have two NADH that are produced. In the citric acid cycle, you have 6 NADH that are produced, right? And 2 FADH2 that are produced, right? During the citric acid cycle, we have 2 ATPs that are produced. Now, the electron transport chain, so when we add up all of this, we have 10 NADH that are produced, and you have 2 FADH2 that are produced. At the end of the day, you have 30 plus 4, right? That's 34, right? But altogether, you have 38 ATPs that are produced, okay? So that's the outcome in terms of energy. Now, when we consider the source of food in our diet, that source of food is diverse, right? We have some proteins, we have some carbohydrates, we have some lipids, fats, oils. All of that can be used as source of energy. But before it can be used as source of energy, they have to first and foremost be broken down into the building blocks, right? Amino acids for proteins, glucose for carbohydrate, glycerol, and fatty acids for lipids. Eventually, all of that is going to be broken down into a metabolic process so that ATP can be produced. Now, based on the source of a food, right, the area where they are going to enter cellular respiration is going to be different, right? Amino acids have a different way to enter cellular respiration for ATP production. At the end of the day, remember that the source of energy is very diverse. That's why you have to diversify your diet, right? So basically, that's what we want to cover in this chapter. Finally, but not least, I want you to remind you to write a little summary for this chapter, right? You can design your own summary. You can go at the end of the chapter and go over the summary at the end of the chapter. Test your knowledge, your understanding of the topic, right? Design your own questions. There are also some questions at the very end of the chapter that can help you there, right? Finally, but not least, create your own handwriting concept maps that can help you also a lot. Thank you for your attention. See you next time.